welcome to Data Science Module 2. Just to give you an introduction on the calendar, this was labeled GitHub and version control. We actually are not going to be covering GitHub and version control this summer since we've decided to switch from Jupyter Notebook, which is a different type of IDE or interactive developer environment, to Google Colab. We have different GitHub availabilities. As you can see on the Google form that I have linked, if you want to, you can learn GitHub tutorials. So there's a link to the GitHub tutorial. There's lots of crash courses. If you would like to learn version control, it's a way of saving and updating your code. It's very useful if you want to get into software development, but is not mandatory to complete this summer course. So if you would like that, I have the resources for that. Nels and I are very happy to talk to you about that. But in order to streamline this course and hopefully get through all of this summer's material, last summer we had to cut off a couple of the courses at the end. We're just going to omit GitHub for today. Don't worry if you don't want to do it. If you do want to do it, please feel free to ask questions. We do have our resources on GitHub. You can access them. It just won't be the method we're using this summer. Okay, so for everyone else, we're going to need to go to Google Colab once again. So we're going to do, you can always just Google Colab. You can type the actual HTTPS Google Colab. We're going to open a new notebook, just like we did last time. I'm going to try and pace myself. I'm also going to try and make less typos this time, but the reality is that probably isn't going to happen. We're going to name this Textile 2023 Data Science Module 2. So the purpose, we're going to add a line of text here. I'm just going to hit text up here. Move this up. I'm going to hit this up arrow so I can rearrange. And I'm going to use this bold because I like bolding things. It's not necessary. The purpose of this is we want to view an image from a computer's perspective or as an array of numbers. Don't worry if you don't know what that means now, you're going to learn. If at any point in these modules you are really getting stuck, I recommend trying for 10 to 15 minutes to Google things and to figure it out yourself. And then once that timer has hit, go ahead and send either me or Nels a message on Slack, and we are really happy to help you. I've given you a lot of introductory knowledge that have hit on a bunch of other things, but we're going to build up there. I don't want at any point for you to feel like this is too hard or you can't do it. So I just want that to be there. Some of you are going to be like way ahead on the coding, in which case you can speed ahead. And when we hit more of that scientific conceptual stuff later, maybe that's where you'll hit a slower roll, or maybe you'll have more to explore. And some of you that might have more bio background but more coding, that might be where you lie. For those of you who have no experience in either, please do not stress. I had never done any coding other than what was required of MATLAB in undergrad until my PhD, and I hadn't done any work with neuroscience until my PhD either. So you're already years ahead of me. So step one is necessary imports. I saw checking the last, oops, I also want this to be text. I just keep doing this to myself. One asterisk. Step one, necessary imports. So I saw during the last module, I hit Shift Enter to make that not look like that, that there were some questions on what is this importing process. So we're going to import NumPy as MP. And so Google Colab, and if you use anything like Anaconda, don't worry if those aren't familiar to you, that's okay. But those of you that are using Anaconda, are Python like interfaces that already have certain packages installed. So Python is the base language, that's the name of it. And then these packages are written in Python to do certain things. So if you look up like NumPy, you can go to the website and it's the numerical Python community and the numerical Python package. So Google Colab has all of NumPy already installed in it. We're just saying here that I want it to also be in my active environment. So because these packages can have quite a lot of data storage to them, we don't want to have them all in at once. But now we're saying, Google Colab, I want you to access this package. So what does a package mean? So a package has a lot of functions. If you want to learn about those functions, you can type like documentation, you can go to a user guide, and you can see everything that they have. You can see NumPy fundamentals, they create arrays, they create numerical structures. We're going to talk about that later. But everything that you need to do all of this work or use something like a function, this is a function, NumPy is already written and it's already optimized. We're just using this like a tool in our toolbox. So Python is like, say, carpentry exists, you can build a house, and NumPy is, say, like a set of hammers that we might use to actually get the work done. When NumPy has a function, this is how it looks. This page is called like user documentation. Don't worry so much about what linspace looks like, but basically it's telling you this is what we've named this function. These are the variables that fit into this function, a starting point, an ending point, the number of numbers, if we need the endpoint as well, and then are we returning it? And then NumPy includes a ton of other information about that. We'll dig more into functions later. You'll have more opportunities to learn from them. The way that I really approach data science is I want you to learn the application. Those of you that are interested in computer science as careers, you will go in your educational backgrounds and you will learn all those details. And those of you that are interested more in the domain side, you'll learn more of the details as you go, but you'll focus on the domain. And that's really okay. It's about learning to balance what you need to know. So all that comes back to NumPy is this package that already exists. And we're just saying, Google Colab, I know you have this. I want to like exist in this. Someone else asked, how do you know what packages Google Colab has installed? I think you can look it up. So we're just going to look at Google Colab Python packages, and you should be able to see every Python library that they have. But if you also need to install other Python libraries like Stack Overflow, this is a fantastic website to go to learn more about coding and to get people's help. You can install any other package that you want as well. So there should be a way to look up what Google Colab already has. NumPy is a pretty basic one. We're also going to import in a different way this time. We're going to say from Skimage import IO. So Skimage is a Python package that's short for scikit image. So scikit image, otherwise known as Skimage, is the scientific kit for image processing in Python. It belongs to a community that's called SciPy, or Scientific Python. That's a community that I belong to. It's a very welcoming community. They have a conference every single year in Austin, Texas. And it's just a really great community of data scientists that tend to be domain scientists. So it's scientific Python, not business or computer scientists. They tend to be engineers or biologists or neuroscientists, and they build this. So scikit image is a package that exists, but this time we're saying from Skimage, I only want you to import this small unit of the, oh my goodness, the unit of functions that exist in there. So the unit is actually an operating systems unit, and we'll see how we use it later. We're going to use a lot of scikit image. I use a lot of scikit image. I've written some tutorials for scikit image. I've talked to the team. They're great. And then the last one, so we haven't used scikit image today, but we are going to. The last one we're going to import is that matplotlib.pyplot as PLT. And so I didn't really introduce matplotlib a ton, so let's go ahead and look into matplotlib. So if you just Google this, matplotlib is a mathematical plotting library for Python, and it does a ton of really basic visuals. What's really nice about matplotlib is you can really control the visual to every single feature if you know how to code. So you can give it the exact colors that you want. If you know how to work with color codes, you can add your labels however you want to. But the visuals that it creates aren't very complex at first. So there's other packages for those of you that are interested, such as Seaborn that I use every once in a while, that you can see gives some more beautiful packages. There's competition in there's competitions in best data visuals that I would love to enter in the future. If any of you are interested in looking into those, I'd be happy to work with you on some of these projects. Nels also does visualization projects. And another big one in Python, I think I just made that up. I don't think it's Atlantean. Python. Okay, I'll get back to you what the big one in visualization is. I haven't had to use it in a while. It starts with an A. But so there's a bunch of different visualization packages. A couple of you asked in the follow up to the last module, how are we going to go from actually visualizing circles on Matplotlib to looking at cells? And we're getting to that today. So I am super excited. So that's the step one of necessary imports. And we'll just hit Shift Enter on this, and you'll see that it ran and that it has a check mark. And so Google Colab did successfully install all of those. The reason that we moved to Google Colab is because it installed all of that and exists in your web browser. There are other ways to do it on your computer yourself using other IDEs and actually downloading the packages to your personal computer. If you are interested in that, chat with me and Nels on our office hours or in an off time, and we're happy to get into that. So the other thing we're going to need to do is we're going to need to push this folder icon over in the left here. And I need you all to upload the image that I attached to this module. So the image is called OGD cells. So the very first time that we published my cell morphology classification was in an OGD model. If you go to the Nance lab and you go to our research and you check out, like if you just control find, oops, you have to go to products. Even I get the website wrong. All of you did a very good job on looking at the website. If you look at Joseph and you find the paper that is Joseph and Lau, and then I'm fourth author, this is actually an image straight from this paper. So this paper is about how we studied nanoparticle and microglia, so the immune cells of the brain, and how they interacted in an ischemic brain. So the ischemic brain is a brain that's cut off from glucose and oxygen, and it causes these cells to be quite stressed. And so we took an image of cells after oxygen glucose deprivation. You can read the paper if you would like to. The PDF is on the website, and that is the image that we're going to import and we're going to work with for the beginning of this class. By the end, we'll get to some different experiments. We'll have entire imaging sets, but for now, we're just going to work with this one image. It's a .tiff file, so .tiffs, these are just good things, I think, to look up. It's not a Toronto file package. Is a tag image file format, so it can either be a TIFF or a TIF. And it's what's called a non-compressed image file format. And so that's really nice because if you convert to something like JPEG, you're actually going to lose some quality from your image. But TIFF is one of the really common imaging modalities we work with in microscopy. So that's important that you have that uploaded into Google Colab. The next thing I'm going to do is I'm going to get back into some coding. I'm going to add a line of text here. I'm going to add an asterisk because I just like italicizing things. You do not have to. And we're going to define something this time, user inputs. This is how I choose to document and how I choose to structure my notebooks. This isn't how you have to do it, but it is typically standard to have all imports for an entire notebook at the top and then to have any major user inputs at the top here. It's just clean, it's easy to read. As you work more and you make your own notebooks, you'll kind of see that. So the one thing we want to define is we want to define some location for our cell image. So I'm naming this variable, which is going to store a location string as cell im location. And then it should just be a period and a forward slash. Actually, it should just be, ignore that. It should just be ogd cells.tiff. Most of the time, I have my exact code in front of me so that I can just read it off and make sure I'm not going to make mistakes. But I'm doing this conversion from Jupyter Notebook to Google Colab Live. And so that's why I stumbled there. But I'm pretty sure that's it. If it's not, we'll get to go through the troubleshooting process together. So that's the only user input that we're going to need for now. And then we're going to add another text line between that. And we're going to label this step 3, reading in the image. Okay, so this time we're going to say the actual cell im is equal to io.imread cell im location. And that gave us a yes. So let me break down what this means. This time we gave our user input a variable for the location of the cell image. So we said that the image is just ogdcells.tiff. And what this means is that it's in the exact same folder that we're currently working in. This time we're saying the actual image, store that actual image information is read in. So what that means is we're taking from Skimage this code imread. So if you look up Skimage imread, it'll give you the Skimage IO documentation. You can control find imread, and it'll tell you this is how you read in an image to a notebook. What we mean by reading in is that this notebook's basically a blank slate. Even though that data exists in a folder, it doesn't exist on this page for us to mess around with. So this is putting that image that we want into the page. An alternative, and I'll just write this here, I'll use an enter, and you don't have to write this code, is if we had wanted to save ourselves that line above, I could have said cell im equals io imread, and then a string, because it's just the name of a cell, ogdcells.tiff. But I like to have this interchangeable so I can really quickly grab a different image. Okay, we're going to add another shift enter to run that. We're going to add another text cell, and this time I'm going to say this is step 4, viewing the image. This is the most exciting part to me. So I'm going to just write out the code first and then we'll go through it. So I'm going to say plt.imshow, image show, open parentheses, cell underscore im, open square bracket 0, comma, colon, C map, the color map, equals gray. And that should work. It didn't work correctly. Okay, so this is not what it's supposed to look like. Let me just do something really quick. Okay, so this is live troubleshooting. Google Colab read my cell image in differently. It swapped my units. So what I'm going to do is I'm going to change this to be colon, 0 and see what happens. Cool. So this time we have actual cells, which is quite exciting. So let me explain to you what has happened now. This is the exact same plotting mechanism we used to make circles before. So this is matplotlib, show me an image, and it's saying read in this cell image that I saved, OGD cells, and give it a gray cell map. But what happened with our cell image shape is that this image actually has multiple colors. Later on in today, I'll show you how to see each of those colors. So each of those colors are saved in a different channel. We use RGB colors. For those of you interested in non-fluorescent immunohistochemistry, which Foon will talk about both fluorescent and non-fluorescent later this summer, their color system is more complex. They don't use RGB, they use I should know this off the top of my head, but they have the pink and the purple. You have to add an extra color deconvolution step. So what I had shown was that I did want the image to be 512 by 512, but I only wanted the 0th dimension because in Python we start counting at 0. So there's 3 dimensions, but there's 0, 1, and 2. And so that's why if I had done it this way, which is saying 0 colon colon, which is only grab the 0th dimension, all other, we get this stick. If I do colon 0 colon, I get this stick because I'm only pulling the first pixel of the 512 row. But if I do colon 0, then I get this entire thing. And so those are actually cells. So now I want you to pause this video and I want you to go to the Google form that I have linked into the Slack message, and I want you to fill out the pre-module. For those of you that are interested, you can also run colon 1, and you can also run colon 2 and see the different color channels. And I'll talk about what those are later. But for everyone, it's just colon 0. Great. So I'm hoping that you've paused and you filled out that beginning pre-module. You can just skip the GitHub portion if you're not interested. And if you want to do it, you can do it on your own time. And we're going to go into this a little bit further. So now I've shown you just how to read an image in, but I want to tell you more about that image. So this is how humans see an image. It might not seem like a lot to you now. There's a bunch of little dots, but I promise you those are something interesting. If we go to channel 2 here, these are our actual microglia, these blobs. They're pretty injured with OGD, and so that's why they look very blobby, because they've been deprived of oxygen and glucose, things that cells need and we need to survive. So these are our actual microglia, which is quite exciting to me. But this is how we view an image. It's not how the computer is going to be viewing or thinking about images. So I'm going to add another text, and I'm going to say step 5, viewing the image the way the computer does. Shift enter. So we already saw earlier that the computer and I had a different idea of how this image was formatted, that I got my dimensions wrong. I thought 3 was first and then 512 and then 512. So this is already an interesting way in which the computer views the image as a 512 by 512 of pixels and 3 different channels of color. But what do I mean by a 512 by 512 of pixels? Well, if I just type in cell M and I hit shift enter, I'm going to see an array of numbers. And so earlier I introduced NumPy, which are numerical arrays. And so arrays are ways of thinking about numbers. If you want to know more about arrays, I highly recommend Khan Academy Linear Algebra. Linear algebra is all about handling arrays, matrices, vectors, and this is basically how images work. They're just arrays of numbers. So what does this mean for us? Well, we can see that there's a lot of parentheses. So how about we simplify this? How about we just take what we're putting into the image, which are those square brackets, colon 0. So when we do that, we're reducing the number of parentheses. Not parentheses, square brackets. So why is that occurring? Because we're only taking this one color channel. Let's change it to 2, and you'll see that those numbers change. So what do the numbers changing mean? Well, every single number in an array is associated with one of these pixels. So at the 0, 0th position, so 0, 0, the value of that pixel is 0. And 0 means completely black. And so the D type we're working with is 16, which is 2 to the 16th. So if you type in 2 to the 16, it's not that high. 2 to the... Okay, the D type we're working with is a 16 unit. but we're only going up to 255. So we're doing 2 to the eighth here. Bits and color work in the eighths. If you want to learn more of that, and you really like this part of it, I highly recommend being an electrical engineer where you learn a lot of signal processing. I know more bio. But so we have 2 to the eighth. We have half of the unit 16 in this image, even though the D type is 16. So our pixel values can go all the way from 0 to 255, because in Python we start counting at 0. So 256 values range from 0 to 255, where 255 would be a completely white point. We don't have a ton of that in this image. This is purposeful. Foon will introduce this more in the future, but our stains are very specific and we get like this very like low amount of imaging. I know that now you can't see a ton, but I promise you, when we learn thresholding in 2 modules from now, so next week, you'll be able to really see the cells and pull a lot out. But we're here just learning things. So I'm just going to go over it again. We're looking at this the way the computer is. This is how the human eye visualizes a bunch of cells, where all of these blobs are actually different, very injured microglia. If we're on channel 0, this is our DAPI stain. And so we can see that the blobs are smaller. DAPI is a nucleus stain. So these are only the nucleus of the cells. They stain DNA. And so that's what you're seeing. That's what you're seeing with DAPI here. We co-stain with both. Let's just go back to 2 because these are my microglia. And so 0, 0th, we're saying there's exactly 0 signal. 0, 1 here, which would be like here, and then right here, we're saying there's a 22 level of signal. And clearly at these points that are much whiter, there's a higher level of signal. This is actually all happening on the green channel on our microscope, but when we import it to a computer, it will automatically create it in grayscale. We can falsely color it if we would like to. If you have more questions about that, please ask. But so this is how a computer views an image. We already talked about cell M shape, but that's just another thing you can learn because I messed up earlier, so we had to learn it earlier. Don't use a parentheses, otherwise it does not work. But now let's talk about actually splitting these by color. So I know that our blue cell M, which is our DAPI, is equal to cell M : 2. No, : 0. I just introduced that to you. Our red cell M is equal to cell _M : 1. And our green cell M is equal to cell M : 2. I know that I told you that this is an RGB, and you're like, well, this is BRG. What are you doing? The confocal that we use can change the order of RGB in which we put the images. So we can choose to image blue and then red and then green. We can choose to just skip red. So you have to have domain knowledge about the image to know which of the channels are a specific color. By the end of the summer, you'll get used to seeing what DAPI looks like and what microglia looks like, where you could kind of guess. But always in these, I talk to the experimentalist, or I took the image myself, and so I know what these dimensions are. So these will change from image to image and from experiment to experiment based on what works with our microscope. Foon and I took videos of her imaging, and she'll actually show you that process, but just some insight into why we might choose one versus the other. Sometimes we need to image one first because a stain is sensitive and we don't want it to be bleached. Or sometimes the confocal will optimize that it's faster to do blue and then red or blue and then green based on where the lasers are located on the machine. Or sometimes we just clicked buttons in a different order. But that's why you might be like, you said it's RGB, but it's BRG. What's happening? You just need to have domain knowledge for the image. So then we've now saved these as their own variables. So if we type in their names, we can see different arrays for each of them. So we can see red cell M, we can see green cell M, and we can see our blue cell M. For you all, I'm just gonna label them here. The ones that matter are microglia and DAPI. If you're interested and you wanna look it up, this is a PI stain, this is a dead cell stain. What did I do? I didn't put a comma in 2 places. I'm gonna fix that. Go back and make sure that you did colon, comma, and a number. I'm not sure if I introduced this. What a colon does is it says for this dimension, I want to take the entirety of it. You can do things with colons where you can do like colon to 5. That'll give you all of the ones up to the 5th one. Or you can do 5 to colon, which will do 5 all the way to the end. But we just want to say all of this dimension, the 0th dimension, the 1st dimension, and the 2nd dimension. Cool. So now you can see that these channels obviously have different values because they're standing for different aspects of cells. But this is just important to know for the purposes of our studies. What's really cool is if we have co-stains, we can quantify those different stains in the same way just by knowing what the stains are. But we need the domain knowledge to know what that biologically means. So I know the greens are actual cells, the blues are nuclei, and PI stains a different aspect of the nuclei when it is a dead cell specifically. Okay, so this time I'm just gonna show the image. Cool. So this time I didn't add those dimensions on it. I said, show me this variable in its entirety. And so what you can see here is an image that has some pretty green pieces. If we go back earlier, you can see that they correlate. But that they're a lot brighter. So what Matplotlib does when you do this is that it's automatically going to threshold your image so that you can see your cells. Thresholding and that concept we introduce next week, so don't worry about it now. I just wanted to preview it for you all here. You can see that that red is that PI stain and that you can almost see no blue. You can see it if you like zoom in, it's overlapping. So what that tells us is that blue is that nuclear stain and it stains all nuclei regardless of whether it's alive or dead. But because I told you that this is OGD, oxygen glucose deprivation, this is our injured cells. Almost all of our cells are dead, which is shown by our microglia being almost completely circular. So anyhow, that's how you can see all the color channels together, or you can plot all the color channels separately. So you can plot IMSHO, red, cell, CMAP equals gray. So why am I saying CMAP equals gray? CMAP, so this just chose the colors chosen by my confocal, so red, green, and blue. CMAP is saying I want it to be gray, and we can barely see anything here, right? You have the wrong channel. Whatever. Looks slightly different than my computer, but we're gonna just move on. I think if I say blue cell, cool. This is the one I wanted to look at. So this is our blue cell elements, our DAPI. If I don't say gray, then it's going to go to the default, which is a color I really don't like. So this doesn't make intuitive sense to me to go to their default color because the background is like this navy. So that's why I always say CMAP equals gray. If you like really want to mess around with colors, you can look up Matplotlib color maps and it'll give you every single color map that they have options for. So I can just type in grays, or I could have changed this to, let's mess around, purples. These are all just things that you can mess around with in your free time. So like the purples look completely different and you can see that they start in a completely white. So if I also did gray, which is what we had earlier where I had CMAP equals gray, black is a value for 0. If I do grays, I never remember how to spell grays. Let me just... They spell grays with an E, but they spell gray with an A. Then the color actually starts with white, and it doesn't actually change the value of the data, but it changes how our brain thinks about the data. So I always go with CMAP gray. I just wanted to give you all of that information. So this is that DAPI, all of our nuclei. But then what if we wanted to view all of them at once? So we're going to use that plotting ability again, where we're going to say some figure with some axes equals PLT, so we're calling that package matplotlib, and we want it to be a subplot of 2 by 2s. You'll see what that means in a second. Last time we didn't do this, but this time we're going to define some axes. So we're going to say an axis 0, 0, which in Python we start counting at 0, so it's in the first position in the column and in the row. We want it to show, so we say im show, that's a function for showing an image, red cell im cmap equals gray. And then for ax1, 0, im show green cell im cmap equals gray. Watch my typos there. And then 0, 1, im show blue cell im cmap equals gray. And then because I like to show it and because I made a cube, ax1, 1, im show that basic cell im that I showed you earlier. Typo. So now you can see them all side by side, and you can kind of see how they make up this cooler image where we have all of our blue, all of our red. PI just has a smaller range of intensities it shows up on. You'll see with thresholding how it immediately becomes much more red, like you can see in this image. So that will be a thing that we'll learn. But yeah, that was kind of our like exciting intro to that today. I am going to show you one more thing. I'll talk about how this works next time, but this time I just want to introduce a little bit more data visualization. So what I'm going to do is I'm going to copy this, I'm going to paste it down here, and I'm going to add myself a few rows below this one. So I'm just going to hit normal enter. And I'm going to steal this axis. So I'm still going to say ax square bracket 0, 0. And I'm going to say this thing get x axis, open parentheses, close parentheses, set visible, false. If I can spell false. And I'm going to do the exact same thing. So ax, open bracket, close bracket, get y axis, set visible, false. So what I'm doing here is I'm removing the x and y axes. You can hit shift enter now. Yeah. You can hit shift enter now. They changed it. This is outdated now. Okay, we're not going to do that. What it used to do is that it would remove the x and y axes. Let's look it up. Matplotlib remove x and y axis. This is in 2011. Let's try these. Let's try this one. This is how you troubleshoot. Oh, I don't like that. I don't think this is gonna work. Yeah, that did work. Okay, fantastic. They've changed their code. So that's how you troubleshoot. Go to Stack Overflow. You say, what are they doing? There were 10 answers that one-upped this. Clearly the older method has been like changed, so the updated version of the package means it doesn't work anymore, but this one's still working, so we're going to go with it. We're just going to copy and paste this, and we're going to say set y-axis, set ticks empty. So what this is doing, if you missed that, is on this first image, so in this 0th, we've removed the x and y axes. I'm looking at an image, I don't care to see them. What I'm going to do is then I'm also going to give it a title, and I hope they haven't also changed how to set title. I feel like this one is more standard. Cool. So now we can see that it's a red channel PI. I'm just teaching you some data viz to make it easier. We're literally going to copy and paste that below each of the other ones. Make our lives easier. There's ways to do this with for loops. If you want to, you can get funky. I'm just going to do it easily. Make sure that all of your axes square brackets now match the correct row, and make sure that the title, this is going to say all channels, goes along with the color that you're doing. So this is blue channel Dappy, if you want to look up Dappy. That's how you spell it. And this is the green channel IBA1. IBA1 is a specific type of microglial stain. So that just looks a little bit better. We're learning data viz. This is a better way to present it. You can mess around with changing fonts if you want to. At this point, this is the end of our module. I have uploaded a different image that has a different shape in to Slack as well. So I challenge you to upload that image. It's just called cells. It has a different magnification on it, to upload that and get it working. But the shape is different here. So you'll have to keep in mind and check the shape. Thank you all for joining me for Data Science Module 2. I'm excited to have you with me for Data Science Module 3 on Wednesday.